Hello guys and welcome back to my channel Mrs.Biology with Neil, and in this video we are going to discuss about the cell structure and its function, and also we'll go in detail about the fluid mosaic model. But first of all, those who are new, do click subscribe button to keep updated with all the latest videos. So today what questions we will aim to answer is, can you describe the structure of cell membrane with reference to the fluid mosaic model? This is the most frequently asked theory question in the AS level. So this is the question which we are going to aim, and in this video whatever I am going to teach you, you can write down the points for the same to answer this question according to the marks given to you. So let's start. So let's first start with all the functions of the cell membrane and also why we need the cell membrane. First of all, let's start with that concept first. So first most function of the cell membrane is it controls the entry and exit of the substances. It is the physical boundary between outside and inside of the cell membrane. It decides what goes in and out of the cell membrane. Therefore, the cell membranes are also known as partially permeable membrane. The cell contains an organelle. That is basically it compartmentalizes individual cell processes because there are many cell organelles present inside the cell. So to give individuality to each of the functions, mainly the cell membranes are really helpful. It basically compartmentalizes. It divides the individual cell processes which are happening inside the cell. That's the second function of the cell membrane. Moving forward, it communicates with the external environment. How do the cell communicate? That is also one of the topics included in cell signaling, which we will discuss in the next videos. So, because of the cell membrane, communication with external environment is possible. That is the cell recognition, how each cell will recognize. Cell signaling, as I said. It allows to change the shape. The shape of the particular cell can be changed according to the cell membranes which are present. It is because of the attachment of the cytoskeleton. The transport and packaging within the cell happens because of the cell membrane. It is site for the chemical reactions. So these are the main functions of the cell membrane, or you can also call it as a plasma membrane. Why do we need the cell? We need the cell to perform all these different functions which are available here. So you can take a moment, pause this video and write down the functions of the cell membrane. Next is, in order to carry out this many functions, they have a structure which are made up of following compartments. Now, in order to carry out this many functions, they have a structure which are made up of following components. They are made up of lipids, they are made up of proteins, they are made up of carbohydrates. The lipid parts are mainly made up of phospholipids and steroids. The protein parts are mainly integral and peripheral protein, and carbohydrates are mainly glycoproteins and glycolipids. So we are going to discuss about each of it in detail. Okay, in lipids where steroids are there, here we are going to discuss about the cholesterols, mainly that how they are helpful in the functionality of the cell. So these are the main composition of the cell membrane or the plasma membrane: lipids, proteins, and carbohydrates. if you want to know the content of each of it, I'll let you know. Lipid is mainly composed of 20 to 40%. Proteins, they are mainly composed of 60 to 80%. And carbohydrates are mainly 2 to 10%. So this is the composition percentage-wise of how the plasma membrane is composed of lipid, proteins, and carbohydrates. So let's first start with the phospholipid. So membranes, they are mainly composed of phospholipids. So this is how the phospholipids are present in the cell membrane. If we zoom in, we'll observe this kind of structure of the phospholipid. This one particular part of the phospholipid has been zoomed in. What all these parts are, this is the phosphate group, the red one. Next up we have the phosphoester bond, phosphate. Next is glycerol. So basically glycerol and phosphate groups, they are bonded with the phosphoester bond. Next up we have the ester bond, and the green part is the fatty acids. So fatty acids and glycerols, they are mainly bonded with the ester bond, and glycerol and phosphates, they are mainly bonded with the phosphoester bond because the phosphate group is present right here in the head part. Phosphate group mainly gives the hydrophilic head component. Because of the presence of the phosphate group, hydrophilic part will be the head part, that is the water loving part. And the tail part will be hydrophobic. They are water hating. Phobic means you have scariness. Basically, you are scared of something. You have the phobia of something. So they have the phobia of hydrogen. Basically, water, hydrophobic tail. So like that, you can remember it on the tip. Yeah, so this is how the phospholipid are arranged. We have the hydrophilic head part, which is mainly made up of phosphate group, and the fatty acid tail part, which is hydrophobic in nature, water hating tail, water loving head, hydrophilic. So this is how it is arranged. You can just draw the structure for the same. If you want, this is the phosphate group, this is the glycerol part, and this are the fatty acids chain. Fatty acids can be saturated, unsaturated. Any unsaturated are the ones which have the double bonds, which have the kings present in it, which are more banded. They are the unsaturated fatty acids. Okay, so you can just take a movement, the structure formula. This is the space filling model for the same, and this is the phospholipid symbol. That's how you symbolize the phospholipid. Take a moment over your and just draw this structure in your notebook because it is very important. Now, since the polar hydrophilic head are water soluble and hydrophobic head are water insoluble, if we put a droplet of phospholipid into a water, it will form a micelle. That is, phospholipid, it forms a micelle when submerged in water. So micelle is like a sphere with head facing outside and tail facing inside, that is away from the water. Our body, it's mainly made up of around 60% of water, and water can be found inside cell, outside the cell, in the bloodstream. So because of the location of the water, the phospholipid hydrophilic and hydrophobic end enables the membrane to form a bilayer of liquid, which looks like this. Like this. It forms a bilayered structure where the phosphate head will be facing an aqueous part Okay, and here also we have the cytoplasm, which is the aqueous part. So they are hydrophilic phosphate heads facing aqueous solution, and inside we have the tails. This is all because of the location of the water, because cytoplasms mainly made up of water. Outside, inside cell bloodstream, they all contain water. So because of this, the phospholipid forms a bilayer. Tail will be away from the water, head will be towards the aqueous solution, towards the water. The currently accepted model of the cell membrane was mainly developed by the scientists Singer and Nicholson in 1972, which is known as fluid mosaic model. So basically, if you want to understand about the structure composition, the fluid mosaic model of plasma membrane is really very helpful. So here if you see, the plasma membrane looks like a sea of lipid with icebergs floating the membrane, which is very flexible. It is described as fluid, that is because both the phospholipids and the proteins which are present, they can move about by the diffusion. You see, like this. Like this. These are the integral proteins, they can move about by the process of diffusion. Therefore, the word fluid has come. So basically, they have the fluidity. The plasma membrane has the fluidity, which is something similar to the olive oils. And phospholipids, they can move sideways, mainly in their own layers. And some of the proteins, which I showed you, they can also move along. And the word mosaic, it comes from the patterns which is produced by the scattered protein molecule when the surface of the membrane is viewed from above. So fluid mainly comes from this flexibility where the proteins can move by the process of diffusion sideways, and mosaic is mainly because of the arrangement of these proteins. When you view this cell membrane from the top, it looks like an mosaic, the patterns of different proteins. So therefore, the fluid mosaic model of the plasma membrane was really helpful to study the fluidity of the membrane. So I have a short animated video for the same, that how the mosaic model, mosaic pattern is seen from the top view, the scattered proteins. Just see to it. It's the cell membrane. So if you see, this is the side view of the cell membrane and this is the top view. So if you see, it looks like a mosaic. How the proteins are scattered in all the direction, it gives a mosaic-like view. Therefore, it is known as mosaic. Fluid is because of the movement of the proteins and phospholipids in their ways. The next component which we have is the proteins. So what is the role of the proteins and what is the function of the protein is fat-soluble organic molecules. They can diffuse through the bilayer, right? But the polar molecules, they requires a protein because the tails, they will hinder the entry of the polar molecule since they are hydrophobic in nature. Whenever they seize the polar molecule, they will just get. So here you see fat-soluble molecule can easily diffuse the yellow one, but the purple one was not able. So they require proteins. So there are two types of proteins which are present, mainly the integral proteins and the peripheral proteins. So if you see right here, these are the integral proteins which are embedded inside the phospholipid layer, and peripheral proteins are present just on the top, in the phosphate head part. So proteins are of two types based on the position, integral and peripheral. Integral means which are embedded in the phospholipid bilayer, and peripheral which are present right there on the head part. So integral proteins, as their name suggested, they are integrated into the membrane. They have at least one hydrophobic region that will anchor them to hydrophobic core of the phospholipid bilayer. So mainly what we are going to discuss in integral membrane proteins are transmembrane proteins, which are extended all the way across the membrane. That is the transmembrane proteins. So transmembrane proteins, like water-soluble substances, they can pass through these protein channels which are present in the bilayer. So these blue ones are water-soluble substances. They cannot directly pass through these tails because they are hydrophobic in nature. So they require these transmembrane proteins. These channels, they can be permanent or temporary depending upon their location. Gated channels. There are gated channels also which allow selected molecules to go through it, whether they are closed or open. So gated channels will mainly be helpful in the action potentials. The sodium gated channels, potassium gated channels are there. So they will only be opened if sodiums are there, if potassiums are there, respectively. Other channels, they are not sensitive to any molecules. They may allow the substances to pass through freely in both the direction, inside and outside. And some channels, they are active carrier. They require ATP, which uses energy to transport substances like sodium, potassium, pump, which we will discuss in detail. Next up, we have the peripheral membrane proteins. They are found on the outside and also inside of the membranes, which are attached either to integral proteins or to the phospholipids. But unlike integral membrane proteins, peripheral proteins do not stick into the hydrophobic core of the membrane. They tend to be more loosely attached because, as the name suggests, they are peripheral proteins. So peripheral proteins, they are glycoproteins if they are present on the glycolipid part, found on the cell surface membrane where protein molecules are attached on the carbohydrate, and they are known as glycoproteins. Okay, when they are attached on the carbohydrate group, they are known as glycoproteins. This allows cell to communicate with one another. They are the one which is responsible for the cell communication, cell signaling process. Individual group of cells, they have their own glycoproteins which are recognizable by immune system. Proteins on the cell surface, they also act as a receiver for incoming messages such as hormones. So basically these proteins are helpful in cell signaling process. Intracellular cell membrane may also contain enzymes for the cell surface reactions. So that's about the peripheral proteins. They are mainly required for cell communication, cell signaling. Third ones are the carbohydrates, that are glycolipids and glycoproteins. So basically, if you see the structure, lipids and protein molecules, they are present on the cell surface membrane, and they have a short chain of carbohydrate. This right here, the red ones which are observed. Present, this short chain of carbohydrate present, this combination molecules are glycolipid and glycoprotein. If these carbohydrates are present directly on the lipids, they are known as glycolipid. If they are present on the proteins, they are known as glycoproteins, which we have discussed in the peripheral protein part. So this red one you observe. So this, it is embedded on the protein. So therefore this is the glycoprotein. This right here is directly embedded on the phosphate head. It is on the phospholipid bilayer. Therefore it is known as glycolipid. So this is the carbohydrate composition of the plasma membrane. Let's discuss how they are helpful. So the carbohydrate chains, they project like an antenna into this watery fluid which is surrounding the cell, where they form a hydrogen bond with the water molecule, and it helps to stabilize the membrane structure. So basically this carbohydrate chain's function is stabilize the membrane structure. The carbohydrate chain, they form a sugary coating to these cells which are known as glycocalyx. In animal cell, the glycocalyx is formed mainly from the glycoproteins. In plant cell, it is mainly comprises of glycolipids. The main function of these carbohydrate chains are as a receptor molecule, which bind with a particular substance at the cell surface. So different cells have different receptors depending on their function. So there are three major group of receptors: signaling receptor, endocytic receptor, binding cell receptor. Okay, so this carbohydrate chains which are present, glycoprotein, glycolipid, they have two main functions. First one was maintaining membrane structure. First one was maintaining membrane structure. And second one is they are mainly responsible for they are basically the cell receptors. They help in cell signaling and cell recognition, cell communications basically. So if we talk about the signaling receptor, they are the part of the signaling system that coordinates the activity of the cell. So this receptor, they recognizes the messengers molecule like hormones and neurotransmitters. So whenever this messenger molecule will bind to the receptor, a series of chemical reactions will be triggered and then the signal will be passed. This we will discuss in the cell signaling video. Second group was endocytic receptors. So they bind to the molecules that are part of the structures to be engulfed by the cell surface. Endocytic means inside. So they will bind to these structures which need to be engulfed by the cell surface membrane. And the third was involved in binding cells to the other cells, that is cell adhesion. Cell to cell communication, they bind to from one cell to another cell, that is cell adhesion in tissues and organs of the cells. These three receptors mainly were present, which is the functions of the glycoproteins and glycolipids. Some glycoproteins and glycolipids, they act as a cell markers also, or antigens, which allows cell to recognize. How cell will recognize each other? So they have the cell markers. So these cell markers are glycoproteins and glycolipids itself. That is, each type of cell has its own identity. They have their own antigen, which is like the different flags of the countries. So these are what? Cell markers are nothing but they are glycoproteins and glycolipids. For example, the ABO blood grouping. Antigens are glycolipids and proteins which have some differences in their carbohydrate portions. So I hope now you can write down the answer for the same. Can you describe the structure of the cell membrane with reference to the fluid mosaic model? So you need to first of all write down the components. According to the components, you need to write down their functions. What are the functions of each of them? That is, what are the functions of the phospholipids, what are the functions of the proteins, integral, peripheral, what are the functions of the carbohydrate, glycolipids and glycoproteins. Steroids, that is cholesterol, we will discuss in detail because they are involved in the fluidity. So our next video is all about the fluidity of the cell membrane. In that we will discuss about the structure of the cholesterol. So in summary, now if you go back, phospholipid is mainly because it gives the semi-permeability of the membrane. So if you check out the function, the first function that controls entry and exit of the substance, partially permeable membrane, it is because of the phospholipid. Okay. Next is compartmentalization, communication with the external environment, the protein composition. They are mainly helpful in communication. Cell recognition, cell signaling, that was mainly because of the carbohydrates, glycoproteins and glycolipids allow cells to change the shape. The proteins are the one which gives the structural integrity to the cell. So proteins has one of the function to that also. That is attachment for the cytoskeleton. Transport and packaging within the cell, site for chemical reactions. So now I hope I am clear with all of this function with respect to its composition, which are this. Okay. So thank you everyone, and if you like this video, do subscribe my channel, hit the like button and stay tuned for the membrane fluidity.